In this video, we will discuss about mechanism of recursive splicing, or we can say the splicing of pre-mRNA so that it becomes mature mRNA and then move to the cytoplasm. So, before going directly into the topic, let us understand some background about this mRNA. So, mRNA, when transcribed from DNA, it comes in the form of intron and exon. And so far, we know that introns are the non-coding sequence, and during this splicing step, the precursor mRNA will uh, go under splicing, so that the introns are removed, and then exons part of the mRNA will join together to form a mature mRNA. Then they will be transported into the cytoplasm, where the translation machinery, that is ribosome, will read this mRNA and forms the sequence of amino acid, or which later on, after post translation modification, becomes a functional protein. So that is, in this video we will discuss some information how this splicing mechanism occurs, or what are the things that we know about this splicing mechanism so far. So this is the precursor RNA or pre-RNA that is transcribed from the DNA, and this blue portion represents the exon, and this red portion represents the intron, and this some small uh, purple and green color represents the sites of the intron. That is, this is the 5 prime sites, purple is the 5 prime sites, and blue is the 3 prime sites. So how this process begins is that first the upstream intron sequence is removed by 3 prime splice sites, and this sequence we can read it from 5 prime to 3 prime. So first, this is the 5 prime site. So first, the first process that occurs is the removal of the intron from the 3 prime splice sites, so that this and 3 prime splice site will remove the intron that is upstream to it. This 3 prime splice site will remove this intron that is upstream to it, and then it will move to the second step. So after first step, mRNA will look like this. So now the second step happens, uh, in, in which the 5 prime splice site will remove the downstream of the intron sequence. So this 5 prime splice site will remove the downstream of the intron sequence, and this will leads to the complete removal of the intron, and this mature. However, there is another condition occurs where there is a presence of recursive splicing, or we can say uh, this RS exon is present. Recursive exon is present in between the introns, and this is not the part of mature mRNA. So this also needs to be removed or taken care of. So how, let us understand how this process happens within the cells. So process is somewhat similar to that we have discussed in the first step. That is, first the 3 primes splice site will remove the upstream intron sequence, and then the precursor mRNA will become like this, and after that there will be the two 5 prime splice site. One is this and another one is this. So in this case, both of these 2 prime splice site will compete each other to remove the downstream intron sequence. If this 5 prime splice site wins, so and then the sequence will become like this. If this 5 prime splice site wins, then this whole part is removed and the sequence become like this. So when the suppose the this splice 5 prime splice site wins and the sequence become like this, so this is not stable, and this mRNA will be degraded by NMD, or the or what we can say, the full form of NMD is nonsense mediated RNA decay. So this process, that is nonsense mediated RNA decay, will degrade the mature RNA that contains this recursive exon. And the other possibility, that is in which the correct exons are uh, joined together after splicing. This will then proceed into the cytoplasm, where it will be read by uh, translation machinery or ribosome, and then forms a sequence of amino acid, and then after post translation it will form a mature functional protein. So in this way, the cell machinery will take care of this recursive exon if it is formed in the mature RNA. And how uh, so from this video you will understand, you have understood how this splicing mechanism works, and how it will take care of the uh, we can say undesired condition uh, that occurred within the cells. So if you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.